Hello. I'm John Barros, and I am going to be presenting my Bio 100 presentation, which is, I think this go. Yes. So the title of my project is Insect Population and Richness versus Microenvironment. Like I said, I am John Barros. I'm in the biology ecology department at school, and of course, I attend the University of Maine. I thought I would start out with some brief introduction and background to the content. Insects, they serve multiple roles in all environments across the globe, except for the Arctic, of course, it's too cold for them. But just for some quick examples, you have worms and beetles that serve as decomposers, breaking down dead organic matter to help nutrients be returned to the soil. You have butterfly and bee species, which help pollinate flowers and aid in the production of wild plants, and larger insects like dragonflies and praying mantises serve to reduce the numbers of mosquitoes and other harmful smaller insects like mosquitoes that spread diseases among humans. Um, when it comes to insects and similar invertebrates, different species prefer to be active at different times. You can have species like bees and butterflies, like I mentioned. They're most active during the day, so they're known as diurnal. Some spiders and moths, they're more active at night, at nocturnal. And then mosquitoes, which are more active during dawn and dusk, they are known as crepuscular. Um, Insects themselves are exothermic, meaning they're cold-blooded, so that their body temperature is more dependent on their surroundings than would say a mammal, which is warm-blooded. And it also means that they would be more or less active depending on said temperature. So in the winter, you'd see a lot less bugs around because it's colder, so their metabolism isn't going as fast. In the bottom right there, those are just two sources that I used for this information, and I will give a full nod to that later. So my outlined problem and hypothesis. So my problem that I first developed, or question rather, is how does the biodiversity of diurnal ground insects, diurnal meaning daytime, and invertebrates differ in different microenvironments, such as well-shaded areas surrounded by coniferous trees, partially shaded areas with rocky soils, it's more deciduous, and sunny, drier areas with a sandy soil, more arid. My hypothesis in the beginning was that I thought that the largest amount of the ground insect biodiversity would be in the coniferous environment, the shaded environment with the coniferous trees, because the shaded area would have the most cover from predators and have the damper soil that allowed for more water access for these insects. For my methods, so in a really brief summary, I had outlined three microenvironments: deciduous, coniferous, and arid. In my pictures, the top right of my pictures is coniferous, the bottom left is deciduous, and the bottom right is arid. Um, I broke out each environment into a equally distanced chart. I drew it out, so I would have 6 zones in each of these, and in each of the 6 zones, I would have 4 sections. Um, I would go into a random number generator and I would select 2 zones and 2 sections from each microenvironment. So, for example, in the coniferous, I had zone 3, section 2, and zone 4, section 1. Um, I did that for each microenvironment. And then once in the field, I located the precise location that I drew out and I placed down my quadrat, which you can see in the top left-hand corner. And for I placed it down, and for around 2.5 minutes, I took note of all the insects that were in and entered the quadrat. Um, some I did know by name, but however, others that I did not know, I entered their details, what they looked like, abdomen size, number of legs, and I recorded it into a insect database for Massachusetts to best identify the species. So what happened? What did I learn from this? So I learned that both the species richness and overall insect biodiversity was higher in the arid microenvironment than the deciduous or coniferous microenvironment. As you can see by the top graph, which indicates the total number of insects, you can see the arid has 12, followed by the deciduous environment with 8 and coniferous with 7. Um, And I found this simply by just counting the total number of insects on the days I recorded data. So 3 bees, 2 beetles, 4 moths, like that, add them all up. Um, and then I calculated species richness. Arid had 8, the deciduous environment had 6, and the coniferous had 3. And this was done by just counting the different types of species. So I could have maybe 5 wasps, but if they're all the same species, that just counts as one. However, if that one species of wasp was found in all 3 of the microenvironments, they would each have 1 in each of the microenvironment. It wouldn't be counted just once for one. So for a conclusion discussion, based on the data collected, it rejects my initial hypothesis, as I thought that the coniferous environment would have the most biodiversity as far as insects, and it turned out the arid showed to have the most insect abundance and species richness. Um, it could be, as I thought, in part due to the weather. Um, as in New England, the days are getting shorter and cooler. Insects are not going to be out in areas that are already cool, that have less sunlight, like the coniferous zone. So the arid zone might actually have provided a better condition for them having more access to sunlight. Um, However, what I do want to know is that the results do have some issues. The deciduous zone was partially cleared by electricians in order to get access to a telephone pole. So the land that I was studying was essentially cleared in order to access the pole, which I believe could have impacted the numbers, as the leaf litter that a lot of the insects would have liked to hide in was cleared out, the mounds that they could have built destroyed. And also really interesting that I found out in my research was that the telephone pole itself, the resin it is coated in to make sure nothing grows on it, is actually coated in a very toxic material that's very bad for insects and animals. And my research actually found out that the toxin is called pentachlorophenol, and it's actually banned in 26 countries due to the neurological damage it causes humans that receive large exposure or ingestion. In the United States, it's only used on telephone poles, but I still find it very interesting that we know the dangers of this chemical and we're still deciding to use it. It also has very bad implications if it gets into the groundwater. Um, what would I want to change about my experiment if I wanted to continue it further? So one of the things I would like to do is conduct it in summer. Um, this class was a fall semester class, so as I continued the experiment, it was kept getting colder and colder. So in the summer, when insect activity is usually at its peak, I wonder if my results would reflect more of what I hypothesized. And I feel that I would want to have the same microenvironments, but maybe a more protected location. I have that image to your right is Massasoit State Park, which is in Massachusetts and about 20 minutes away from where I live. I think it'd be better to have a more protected area that's not as involved with humans. In Massasoit, you really don't have on the trails a lot of human activity as far as repairing and clearing of area. If you have walking trails, but if you stay to the side to the outskirts of it, you'll have a more remote area, which I think could provide more unbiased data. And as far as other ideas for the future on what I would like to do, I would like to observe different plant or fungi accumulations in these environments. So the same sort of process, maybe not quadrat sampling, but perhaps doing a large area or transect and seeing how fungi or plant accumulations differ. Um, I'd like to see how the length differentiates for insects to return to normal levels after a disturbance. So let's say the moving of fallen debris or rainfall like we saw in the clearing. I think it'd be interesting to see how long it takes for certain species to come back. Like let's say ants that are known to walk around and monitor different locations of the environment versus an insect that, like a spider, that has a particular burrow. Let's see how that would alter. And I would also like to see how insect levels change in relation to distance from a telephone pole, sort of transect experiment, as I believe I want to see how these toxins affect, if they affect at all, the accumulation of insects around that area. Finally, here are my works cited that I had for this experiment, as well as my acknowledgments. I would like to thank my TA, Elise. She helped me so much with this process. I've probably sent her revisions like 10, 15 times, and every time she gets right back to me, and she's amazing. Dr. McGuire, I've sent her also numerous questions and revision tips, and she's always been able to help me. And also my fellow peer, Elizabeth, she and I have worked together extensively doing peer edits and reviews of each other's work, and I really don't think that my work would reflect the same if not for all of their help. So I truly appreciate it. All right, so that has been my, see if I can stop screen sharing. There we go. So yeah, that has been my report. Um, I wanted to highlight my report on all of the topics as I believed that I didn't find one point more interesting than the other. I thought the whole process was very interesting, and I was really happy doing all of it. I think that the biggest thing that I personally learned from all of this was just the great flexibility you have with all of this stuff. Like when I did more formal lab reports in high school, it was very structured, like do this now, do that, do that. You don't have a lot of options. But for here, I got to choose how I wanted to do everything. Um, I could choose if I want to do transect, quadrat, random sampling. I could do anything I really wanted to. I didn't have to just sit in one room, record same thing over and over and over again. I went out different days, different types of weather going on, different conditions every single day, and I thought it was really interesting. I thought it was really um, beneficial to understand that in like a real life scenario that you have control over what you want to do. There's not there is a sort of a structure you have to follow, but it's not extremely rigorous. You can choose what path you'd want to go, and I think that's really beneficial for people to know. So thank you for listening, and have a great day. Goodbye.